So, so here is the broad story or the snapshot of this story, which is that we all originated somewhere here and stayed here for a very long period of time. As we understand that because we stayed here for so long, there has been huge amount of genetic diversity during that period of time, and we moved with a very small amount, a very small fraction of the genetic diversity to outside of this world. It was primarily here, and there's evidences coming from very strange quarters, which if I get time, I'll also discuss, that we were all together somewhere primarily here and then moved to different directions. One direction very early went here, and if you imagine that going to Australia would require a lot of this, that's probably not true, because when this happened, it was Ice Age. And Ice Age means that there was a lot of this water in the ocean which was actually trapped in the glaciers. All right, and which means that the land bridge or continuous land bridge continued from here till up to the tip of what we call today's Bali. All right, so that it was, people could actually walk along the coastline to this place. And then eventually there is something which is across this called the Wallace line. Remember Wallace, who was Darwin's co-companion in that beagle? So Wallace line, which means that was a separation between Australia and the rest of the world. How do we know this? Because if you look at the species of birds and beasts in Australia, they're very different from what you find in the other parts of the world, which means that there was no automatic, particularly for land animals to move from Australia, or there was no easy passage between Australia and the rest of this world. And that's why there is a hypothetical line called Wallace line somewhere here, which means that there was a huge separation between these two land masses by sea for a very long period of time. So the Wallace line essentially passes through this, which means that eventually anatomically modern humans, when they moved here, they had to cross this. They had to set sail for a completely unknown land which they even cannot see. Because even if by the best estimates, the tip, if you get to here, you still cannot see Australia. So they did set sail to something which is completely unknown in the sea. But by the time they have moved to all these places, which means they might have understood or developed crafting of boats, most likely because they were living around the seashore for such a long period of time. This gives us another interesting twist to this. If people have really moved through these coastlines, right? So those coastlines which were there, right now, after the ices have all melted, are right now underwater, right? So if you are trying to build a model, or if you're trying to even test the hypothesis that whether this kind of movement at all happened, you really do not have an archaeological handle on this. You cannot ask an archaeologist to go and excavate under the sea and try and find out remnants of a civilization or something from that. So you are only, this is another advantage of doing it in a molecular way. You can only use genetics, or you can use genetics in a much more efficient way in trying figuring out the history which might have happened at around that time. All right, so this is mitochondria. As we have mentioned, that mitochondria has two kinds of hands on it. One is a fast teaching hand where mutations accumulate pretty rapidly. Another is a small teaching hand where mutations do not accumulate rapidly. So depending on the slow occurring mutations, mitochondria are defined into haplogroups, which means that if a haplogroup is basically very, very stable, and on a haplogroup background, there are many, many haplotypes which have arose. So if you look into the haplotype, so this happened much later. This happened much later, and this is another way to strongly corroborate the story that has been built. This is the story of three sisters or three haplogroups. So if you look into, this is again a coalescent tree like this, using mitochondria, and if you look into this, then you find that there are ancient haplogroups which people have named as L1 and L2, which you find only in individuals who are in Africa today. So the L1 and L2 haplogroups, even today, are found only in individuals who originate somewhere from Africa. And it's not found anywhere else in the world. Again, I mean, when people found this later in time, people were not terribly surprised because people now know, I mean, by the time this was happening, people now know that this is where this is what the cradle of civilization has been. So if you take this L1 and L2 and do a back calculation of time, just like this, using the molecular clock, etc., then eventually you come to the coalescent time, which is 192,000 years. So as you can see, it's getting refined. It started with 200,000 years in the first paper. It get down to 192,000 years. The basic story, however, otherwise remains same, that all Africans coalesce here, and eventually all non-Africans coalesce somewhere here, which is about half the time compared to what we have here. Right? So the interesting advantage that we suddenly got, I mean, this is by chance that we got this, is that there was a haplogroup called L3, which is very similar to another haplogroup which we define as a M haplogroup. And this M haplogroup is extremely widespread all across the world. So there are two other haplogroups called the M and the N, which are very widespread across the world. So this gives you a sense that maybe if these are people who are ancestors to all people of the world, then maybe they have started with just, either they have started with just the M and the N, or the M and the N arose right after people moved out of Africa, right? I mean, we probably cannot test this conclusively, but either one of these must have happened. And this is also testimonial to the fact that it probably started with very small number of individuals. That's the reason that there is not too much of ancient variability here. This is like an ancient variability, and there is not too much of ancient variability here. Whereas in Africa, you see a lot of ancient variabilities. You see L1s and L2s, and the L1s are extremely varied, the L2s are extremely varied within themselves, and so on and so forth. The aim across the world is somehow much more similar compared to anything else. So aim is very similar, N is also very similar, and if you look into this, this is again testimonial to the fact that these are people who are from the Australians. So these are Australian Aboriginals. So if Australian Aboriginals have both M and the N, and they have gone to Africa, gone to Australia, there is very little chance that they would be rapidly moving back and forth and exchanging this. Then very likely that by the time people have moved to Australia, they have already had this M and the N. So again, testimonial of the fact that maybe either we moved with a very small proportion of M and N out of Africa, and then rapidly expanded and moved to different places, and that's the reason everybody else is an M and N. It's very similar to the L3, so that's one possibility. The other possibility is the M and the N arose very rapidly when people were somewhere right after moving out of Africa, and that's where it arose, and that's why it's probably ancestral to many populations all across the world. These are the Europeans. As you can see, the Europeans coalesce much recent in time compared to where the Australian Aboriginals coalesce. The Australian Aborigines coalesce very deep in time in terms of this, and this is estimated to be something around 72,000 years, and this is about 67,000 years. Okay, this individual was an Indian. This was an Indian individual who was included in this particular study. All right. So right after this, there was a paper which came out again in Nature in 1999, which found that in Ethiopia, people thought there is no M haplogroup in the whole of Africa. They found an M haplogroup in a population in Ethiopia, and this is a very different kind of an M, because the other kinds of changes that you see in the mitochondria are very different. And people started postulating that maybe this is how it has moved in, and this is where they stayed for a very long period of time and moved to different directions. One of them, following the coastal route, also went to Australia, and that's the reason that you see M in Australia as well. All right, so people started redrawing this, that they started saying something which is called this is the most plausible northern exit route. Then there was a southern exit route, which is a movement out of this. following the southern route into this. Okay, so right at this point of time, I would like to take a pause and try to revisit the whole concept of recombination. All of this was easy, easy because the entire mitochondria is like a molecule which is passed on from mothers to the offsprings just in toto. Similarly, it was true for the Y chromosome. Now, what happens if you have recombination? The problem with recombination is that I have a chromosome, right? The problem, my chromosome is whatever, I mean, whichever chromosome I choose except the Y or X, whichever chromosome I choose, that chromosome is not entirely derived from my father or my mother. It's like bits and pieces, it's like a mosaic of parts of my father and parts of my mother, and that's because of recombinations happening. Because recombination makes at points and just swaps material and gets stitched and gets passed on, so one of the chromosomes gets passed on, but that chromosome is always already a mosaic between multiple things. So, I'm sorry, my chromosomes are like that, so the chromosome that I would pass is going to be a mosaic of my father and mother's chromosome, right? So here is the problem, that earlier what we could do is, if you have an individual, you had an ancestor, right? One individual had one ancestor, and if you go back, that individual also had one ancestor, and sometimes two individuals can also have one ancestor. So it was only going to decrease. The tree is you start with the tree of size n, it can remain size n or it can decrease in subsequent generations as you go back in the past. Here is a problem when it can go the other way around as well, because my chromosome is a mosaic of my ancestors, and that's the reason that this, if you go back to the ancestors for that particular chromosome, that can come from multiple individuals. All right. So if that is clear to you, so this is how it's happening. So this is the chromosome that the offspring has. It has predominantly, say, red and a little bit of blue. So the red and the blue has come from two different individuals. So this particular chromosome do not have a single ancestor. This particular chromosome has ancestors from two different individuals. So if you now consider sampling chromosomes or sampling individuals, which eventually would mean that you are sampling chromosomes, these individuals will have ancestors who can have multiple ancestors. So the tree can have common ancestors as well, because this part of the chromosome and this part of the chromosome, these two individuals can have a common ancestor as well, because this individual. So suppose this is a chromosome of this individual, and a part of this gets to this individual. This part gets to this individual, and this part again gets to this individual. This part gets to this individual. So if you are starting with these two individuals, it can sometimes go back to this, which is a common ancestral event, but in many cases it will go to two different ancestors, right? So your tree now can go both ways. It can expand, it can increase in the number of ancestors, it can also shrink in the number of ancestors. Now, if you do the maths, it can be shown that eventually it will go back to a common ancestor. If we have a common ancestry, it's still a tractable problem. Under most kinds of real parameters, it eventually goes back to common ancestor. Like, I mean, however it happens, if you take the pieces, each piece will eventually go back to common ancestor. And because your genome is large, you can also think about the genome to be very, very small pieces where there has been no recombination between them, right? So given a small piece, that has a history where there is no recombination happening between them in any time in history. So this is the consequence of recombination, which makes this problem much more complicated, and people have generally tried, and this is not a tree. This is like a graph. This is called an ancestral recombination graph, because now it goes to all directions. It can build networks. It's not like a bifurcating tree like this. All right. So people have. These are not very much, you have to make many approximations in order to try and estimate this. And the other thing people have done is they have refrained from doing that too much, because it has, I'll come back to this, that when we are again started doing this, but we have refrained from doing that too much. Whatever we have been doing is much looking at the diversity of the entire chromosome and looking at how diversity is clustered in different populations. All right. So this is a simple model of population expansion, population evolution. So you start with a few individuals, then there are a few more individuals, and this gives rise to diversity. The allele frequencies changes as the populations expand. When populations expand, it generally puts pressure on the environmental niche, which means that some people would go back and form a new population. When you go and form a new population, you generally only have a smaller subset of the diversity that you have in the parent population, right? That's only natural. That is a small population. It also has a lower diversity. So what is interesting over time is that this population is a dynamic entity. This population is also a dynamic entity, and they all develop signatures of their own. So if they're reproductively isolated, they do not exchange genes, then the patterns in which mutations accumulate in populations, they are very, very different, right? So the patterns in which mutations accumulate in populations, they might be different, and they might give rise to signatures. So if we look into this can give rise to completely distinct populations. So we can just schematically, in a simplified way, we can think about this. This population changes a little bit here, and it starts and it bifurcates into two populations. It does not necessarily need to bifurcate, but whatever it is, and then each of them can give rise to a completely new kind of a signature of a population. So the question that people start, okay, and there is some amount of genetic exchange between them, and so on and so forth. So the question that people started asking is that if that be so, can we, just using the genetic data, agnostically identify individuals from their original populations? So the pattern in which mutations accumulate in these populations, from that, can I take an individual's genome and try and identify from which population the individual has come from? All right? So that's the kind of question that people have started asking. Now, life has become, if this is true, if I can assign ancestors to individuals, and if I can assign ancestral to small segments of chromosomes, that this part of the chromosome has come from this population, or so on and so forth, then right now you can imagine a scenario like this, that there are these four populations, not populations per se, just imagine these are like proxies of populations which have their own signatures, which are differentiated. Is there anybody who is colorblind here? Can all of you see that there are four distinct colors here? Okay, great. Yeah, so suppose the colors stand for a signature of a population, and this individual has two chromosomes which are like this. This individual is like somebody from here and somebody from here has mated to give rise to an individual, right? So that's why this individual's one chromosome is like a population here. This individual's and another chromosome is like a populations here. So this individual will be an admixed individual, admixed between these two populations. Similarly, you can have an individual like this who has, say, 1/4 ancestry from here, 1/4 ancestry from here, and half of the ancestry from here, right? So this means that one of the four grandparents is from this population, one of the four grandparents is from this population, and two of the other grandparents are from this population. So if that be so, then this is this. You can also have complicated mosaics like this and so forth. So why is this important? Because there are populations that we know right now who are admixed between populations which has been separated for a very long period of time. Can we do this? So this is the question that we ask. The other question that we also ask is that can we partition the variability? We see a huge amount of variability, right, in individual. And we want to have a quantitative handle on this. And we try and relate that to the genetic variability that we have. So if we have genetic variability, this is a question which is often asked, that this is again related to social science. What people have asked is that this group of individuals are very different from this group of individuals. So whether there is a genetic basis of that. So in terms of apportionment of human diversity, there was efforts very, very early on by a person, geneticist called Leontief from Harvard. And what Leontief tried to do is he has sampled individuals from different populations all across the world, and these are words which were used in those days. These are seven races, which means populations from different parts of the world. Essentially, these are populations from Pacific Islands, Australian Aboriginals, Europeans, people from Antarctica, people from the Arctic, close to the Arctic Circle, people from all over Africa, and so on and so forth. And he looked into 17 genes. And what he did is something like, something very similar to an analysis of variance kind of a thing, and try to answer the question that how much of this variation is because of variation within a population and how much is because of variation between populations. And this is a 1972 paper, and what Leontief's inferences is that the within population variation is about 85.4%, which means that of the enormous variability that we have in human populations all across the world, most of the variation is variation between individuals, right? It does not depend on which population you sample them from. About 85% of the variation is just variation between two randomly chosen individuals. There is somehow a little bit which is systematic, and about 6.3% is because of continental differences, right? It's the systematic variation between populations which are from different continents. So this is Leontief's work, 1972. So the question okay, so if this is with just 17 genes, and it's 1972, the technology is very, very primitive. This is even painstaking to generate the data on the 72 genes, all right? So we move forward to a time point which is about 2002, and in 2002 it was pretty easy to have 377 microsites, so which is essentially like 377 positions on the genome which was marked on 1056 individuals from 52 populations all across the world, all right? And people are trying to ask that if you are given this data without looking at the population level, can you mathematically cluster these individuals? Or if you cluster them, what would they look like? So remember, there is something around a 6% variability between races, which means that between groups there is still something around a 6% variability. So whether that was enough to actually cluster individuals in different groups. And the way to read this figure is something like this. So there are 1056 individuals. So imagine this is like 1056 vertical lines, all right? So each line is like a genome of an individual, all right? What the program is trying to do is that it's just looking at the data of the individual's genome and trying to, based on some kind of a similarity, it's putting coloring the genomes of individuals or regions of the genomes of individuals same if they're similar, all right? It's not based on one single position, but it's based on a local region around a single position. So if they're completely agnostic of this and tell the program that suppose the 52 populations of the world is divided into two groups, all right? So k equal to 2. Suppose there are two hypothetical populations in the world. What will happen? So what happens is exactly what is expected. There are two groups. One group is essentially here, and another group is essentially here, and it's like a divide in according to the way in which people have moved out. If you look at it, if you do it into three groups, the Africans are separated here, all right? So here are Africans, here are Europeans and Asians and all of them together, and these are people who are from Pacific Islands primarily. All of these small Pacific Island groups are somewhere here. And then there is an optimization method in which they would ask the question that what's the most optimal number of populations in which, it's not the best method, but it's one of the ways in which they come up with. So the estimated number of populations. So if you're clustering this clustering individuals into different populations, when to stop? When do you say that the continental, in terms of continental divide, there are k major populations, or 5 major populations, or 6 major populations? So when they did this, they came up with 5 as the as the best time to stop, and this is how the figure looks like. So imagine there are vertical lines here, which means that there are many Africans here who are colored here in this yellow chrome. Then there are the yellow, this blue, cyan. These are individuals from Europe and Asia all clubbed together. So it cannot separate between Europe and Asia. These are people from East Asia. So these are people from Pakistan and Middle East and all of Europe. These are people from East Asia, Chinese, primarily Chinese. And then there are these individuals who are from Pacific Islands, and these are 2 Aboriginal people from Latin America. All right. So these are the 5 distinct clusters that they found: Native Americans, Pacific Islanders, East Asians, Eurasians generally, and Africans. So this is what? This is 2002, 377, one kind of a marker, which is pretty informative. And they also did this between population, within population proportion, and found very similar to something like a 5%, 5.4% among populations within regions, which is very similar to what people found, what Leontin found using just 17 genes. All right, so the same group, almost the same set of individuals, a few years later down the line, 6 years down the line, now we have a technology to look into 650,000 autosomal SNPs on the genome, right? So you can, it was 377 a few years back on the autosomal chromosomes. Now you have 650,000 autosomal SNPs. SNPs are single nucleotide polymorphisms, that's the technical term, essentially looking at variation in the autosomal part of the genome. And you ask the same question and try to do that. And now you see that this obviously you have much more information here, and now instead of 5, 7 is the best cluster. All right, the Africans, again it's the same kind of picture. Imagine this has 956 vertical lines, each line pertain to an individual. So the Africans stand out here as a group. The Middle Easterns are here, who have this brown color, and obviously the Middle Easterns, as you can see, are admixed between multiple ancestral groups. They have something which is very similar to brown. They have something which is similar to the European ancestry. There's something which is similar to the Central South Asian ancestry. So these are people who are from Palestine, Israel. They're different tribes who live there, Druze and Bedouins and Palestinians, and they are like admixed between all of this. Again, goes back to that story that somewhere there, that's a hot seat of human evolution where people have moved around admixed for a very long period of time. Then there are these Europeans here, again, very distinct, the Central South Asians, the East Asians in this yellow. These are Oceania, these are people from Pacific Islanders, Melanesians and Polynesians, and these are people from Native America, again, in different color codes. If you look at this into a branch, they also use this as a distance matrix and eventually put this in a branch. You again see, I mean, they branch according to their color as well. The populations branch according to their color. All right, so when they're using autosomal data to partition their variants, it was working great, and you can agnostically, without even a knowledge of which individual has come from which, when you cluster and then go back and look at their levels, you see that it goes to continental clusters. So in spite of the fact that there is only about 5% systematic variation between this, that 5% was enough to actually cluster them into different groups. All right, so this is how it worked. So there were 7, so when you refine, there are 7 major clusters, and its geography is playing the most important role, and there has been a huge amount of admixture between different populations, and there is evidence of many populations which are admixed between 2 or 3 or even multiple populations. And geography is correlating wonderfully well with them. So when you have individuals from one region, they're very similar. When you consider a different region which is hugely different, it correlates with the distance from here to here. So there, somebody later has done this even more elegant study, I would imagine, where they have actually looked at, suppose you move out of Africa and move to different directions, all right? Then depending on the length that you cover, and you look at the genetic distance for any two individuals, and you also calculate that if these are the trajectories that people have taken, then what is the difference, or what is the distance of you compared to an African from where it's all started. So you can compare between any two individuals, because you can get the relative distances from one central starting point, and they have looked at that, and they have looked at the number of and the distance from that particular point from which it started, and looked at the correlation of that. So they have looked at the genetic distance versus the geographical distance correlation, and the correlation is as high as close to 80.8. So geography is an amazing correlate in terms of if this kind of movement is if this is the movement, this is the way the movement has happened, and it correlates to 0.8. If you move the center, if you move the epicenter of the starting point from the south central part of Africa to any other continent, then that correlation drops. So just another way of looking at that, it probably all started from Africa and moved to different directions. And our understanding of the directions, the directionalities are also probably pretty robust. All right, so if you look at the proportion of human diversity, appropriate diversity, it now changes a little bit, but it's still less than 10%. So the systematic diversity between populations, again, this is a biased sampling of the 650,000 positions on the autosome, and it's about 10%. Now, if you look at the entire genome, if you are capable of looking at the entire genome, this is again going to go down. This is slightly artificially upwardly biased, and we know why this is. So at the most, it can never, if you even consider global populations, the systematic variation can never exceed something around 10%. So this is how it has moved, and every time it has moved, it has given rise to signatures to populations, because when you move to a new place, you settle there's some kind of and mutations accumulate on it. You interact with your environment, you interact with the people around you, and you get a new genomic signature of your own. And these are discernible. So right now the state of the art is like this. You don't even need to give an individual the entire chromosome. You just give a small portion of your chromosome. And just by looking at that, I can assign that this part of the chromosome has an origin somewhere here in the world, right? Or this individual is most likely from this particular population in the world. And you might be knowing that there are companies which do this with a fee. So they tell people what their ancestries are from, where you are actually from. Yeah. So this is particularly popular among people from United States who have African ancestry, right? The African Americans, because they are very keen to know which part in Africa they might have their ancestors from, and for some of these it can be traced back to a very, very small region. Though we should also appreciate that our genomes are just not from one ancestor, right? It's not from one ancestral position. It's from many, many ancestors put together. So what you're tracing back is probably where most ancestors are, but you still have a lot of this from many, many other regions of, even within Africa and so on and so forth. Okay, so far so good. So this is how it looks like. The yellow chrome is how we have all moved out and moved to different places. And if you look into this map carefully, this is a hypothetical map when there is a lot of these glaciers here. The glaciers are melted, and because there is glacier here, if you look at the coastline is a little different from the coastline that we have in today's maps. So the brown at the coastline, as you can see, there is a very little difference between Australia and the rest of the world, and this is entirely connected. And people have moved into this, and the time estimated from genetic data also correlates very strongly with the time when the first divide between this glacier has happened. And that is estimated using some other kinds of methods, many other kinds of methods. Geologists do that, and we know that the first time, there is a tunnel by which if you enter this, you can move southward, is very similar to the time estimated by using genetic methods to estimate the migration dates of movement into this part of the world. So yeah, I mean, there are these small stories which also put together, and if you put together the small stories, there is this big picture which comes into place. All right. The other thing which I would like to mention here as a passing mention is when people moved out, the world was not like a vacuum. It's not like we are moving into a vacuum space. When people moved out of this, there were many people who were very, very successful as hominids like Neanderthals who were living here, right? They were very, very widespread in Europe. And I'll, if we get time, I'll probably talk a little bit about, hey, how much time do I have?
You have roughly
45 minutes?
40.
Is it 1 o'clock?
Yeah.
Okay. Okay. So there are these hominids who used to live here. All right. And there were hominids probably somewhere else as well. There are mathematical ways in which people have estimated that there were probably also hominids living in Africa when we were in Africa. So we are contemporary to many of them. We are surely contemporary with Neanderthals when Neanderthals were living, were very widespread across Europe. Okay, so I take a departure here, change gears and move to something else, and I start narrating a personal experience of mine. This is to give you a sense of how things have moved. I'll get back to what we were discussing, but so this is about my PhD. When I was doing my PhD, we looked at 58 DNA markers. We looked at mitochondria, Y chromosome, and a few of them autosomal. We also sequenced the mitochondrial hypervariable sequence, all right? And we looked into variation in 44 geographically, linguistically, and socially disparate ethnic groups in India. And we used, so this is what the analysis of this, and we used to proudly proclaim that this to date is one of the largest, if not the largest, genetic data set on Indian population. All right, so this is 2000, 2003. All right, this paper came out in 2003. 2015. So 2003, 2015, 12 years gap. This is 54 DNA markers on 3.2 billion. All right. In 12 years, we have something which is called a 1,000 genome project, where people have done the whole genome sequencing of 2,504 individuals from 26 populations. So this is 3.2 billion into 2,504 individuals. So we had 54 markers in whatever the number of individuals we had. This is 3.2 billion in 2,504. So this is how the field has changed in 12 years. Right? We cannot even calculate the fold difference. And this came up in this particular paper. Another example. So we discussed about the Human Genome Project, right? 500 bases of sequencing and then putting it all together, and 15 years of painstaking effort to come up with the first draft of 13 years of painstaking effort to come up with the first draft, which cost 2.7 billion USD. So the first human reference sequence. This is my institute, and this is again dated. This is 2016. Our institute came up in 2009, and we have something called a genome clock, which means that we do sequencing of humans, and if you put them together in an equivalence term, we have already done sequencing of 8,875 individuals. So just imagine, this is the whole world put together. All universities of all the world, all leading universities put together, they took 15 years to do one genome. And in only this period of time, we had done 8,875 genomes, complete human genomes. So this is how the full field has really moved. This is how the technology has really changed. And that's the reason the whole entire discipline has moved from a slightly biology, molecular biologist paradigm to completely data science paradigm. So it's huge information and data, and from which you need to make sense of it. All right, so how is this quantum leap even possible? So if any of you are from computer science background, you might be knowing about something called a Moore's law. So the Moore's law is essentially an empirical observation, but people found it to be so robust over time that people thought people named it as a law. So what Moore's law states is simple, which is very simple, which is that the power of a transmitter or a semiconductor, that doubles every 2 years, right? So if you have 4 semiconductors in 1 square unit area, one semiconductor in 1 square unit area, then in 2 years' time, you can have 4 of them, which means that that's the rate at which the power of computation increases, that's the rate at which your mobiles become smaller and more powerful compared to the computers that you have, and eventually that's the rate in which it can proceed. And it has been very constant over time. That's why Moore's law is almost like a straight line. That's the reason they have called this as a law. But if you compare that with the sequencing technology, which means that if this is what it is, then Moore's law would also mean that the cost of computing will linearly decrease, right? Because your power is increasing, the cost of computing will linearly decrease. So this is how the cost has decreased. If you compare that with sequencing cost, this is how it was going. At some point of time in 2007 onwards, it has really taken a dip. So the sequencing cost right now is like this. So the first human genome sequence cost the world 2.7 billion USDs. Now a human genome sequence, the entire human genome sequence can be done in less than $1,000. So this is how the dip has really occurred. And not only that, that's only just in price. But if you just consider the time, if you just consider the effort, then the cost is enormous. If you bake that into that cost, then that's just enormous. It's not just a comparison between $1,000 US and 2.7 billion USDs. It's also just imagine so many people spending so much manpower and being paid for so many years, right? Okay, so this is how the technology has moved, which means that this is the rate at which we are sequencing data. This is how it has come down, which means that we are sequencing data at this scale. All right? So obviously the field has moved, and we put all of these findings in test. And what can we do now? So right now, what can we do is something as simple as this. You take one specific individual, all right? If you take just one specific individual, that individual, because of recombination, small parts of your genome belong to different ancestors, right, back in time. So if your genome essentially is bits and pieces from different ancestors. So if you just think about this in a linear way, I have one chromosome. A chromosome has 2 copies, one from my father, one from my mother. If I just fix my attention to a very small region, then this region will have a common ancestor somewhere back in time, right? So if I take a subsequent region, if I take so I do that for a small region, then I move to a next region. I mean, whatever. I do not need to pre-assign the length of the region, but if I start moving in a linear scale, then there is a small region later which will have 2 common ancestors who are completely different. So my genome is a mosaic of individuals contributing their genomes from different place, different time in space and history in the past. And if I compare between two specific homologous region, and I try and go back, then they have common ancestors at different time points. So just by looking at a single individual's genome, I can get a sense of the population from which I have come. How have the common ancestries changed over time? So this is something like this. Suppose, so you recall in your mind that this is something which depends on the population size changes, all right? Okay. So now just imagine something like this. So suppose here is my genome, and I'm just concentrating on this particular region on my genome, right? So I'm concentrating this particular region on my genome, and this region is suppose has come from my father, this has come from my mother. That I know, but if you want to go back to the common ancestor for this particular region, their common ancestor suppose goes back somewhere here. So here there was one individual who has eventually contributed this part of the genome, this part of my genome, which has gone through one trajectory through my mother and come to me. It has come through another trajectory through my, and eventually through my father and come to me, right? This is a very small portion. So for the next portion of the genome, that necessarily does not go back to the same common ancestor. It goes back to a common ancestor who may be farther back in time, right? Now, if I am from a population which has gone through a history like this, so the population has been small, then it has expanded, and then it has come down again, then there will be many more people here in time, back in time. So time is going in this direction. There will be many more people in the population at this point of time in history. So there are many more people who will be contributing parts of their genome to my genome from this part of this interval of history, compared to this particular region of time when there are very few individuals, right? So if there are few individuals, there are fewer individuals to contribute. If there are more individuals, there are more individuals to contribute. So even my data, the data in my single individual's genome is testimonial of how the history of this population from which this individual belongs, how it has gone through that. So what kind of history this population has experienced. So whether we can so this is conceptual. If you can conceptually think about this, then you can try and see whether that happens in practice. And that's what people try to do. So what will happen? What is the cardinal sign of how can you differentiate? So if this individual, if this is a common ancestral segment which I have got through two different trajectories to this to the present time point, and this is much closer in time compared to present compared to this, then what will happen is this particular segment will accumulate more mutation. Is that clear? Right? So this segment will have more mutation here, whereas this segment will have fewer number of mutations. So the whole idea is like this, that if you're scanning over the genome, if you're just linearly scanning over the genome, then if there is a certain region where the parental chromosomes do not differ too much, that would mean that they have a common ancestor who is not too far back in time. If a similar region of the genome, the parental chromosomes differ hugely, that would mean that their common ancestor is much further back in time, right? And this is proportional to how the history of this population has been through. So this is, again, I mean, all of you, any of you or all of you who know hidden Markov model, this essentially fits into a hidden Markov model kind of a framework. It's linear in this, there are different states, and if you apply the algorithm, you can try and find out where these transitions happen. Where is this excess of heterozygosity or excess of difference between the parental chromosomes, which calls you for a ancestor who goes farther back in time. So for this region, there is not too much differences, so the ancestor is not too far back in time. Then suddenly in this region, there is accumulation of many differences. If you do a hidden Markov model, it will tell you that this goes farther back in time, right? So in that way, you can do this. And
what is the exact role of hidden Markov modeling here?
What is the exact role?
What place?
So the hidden Markov model is doing, so this is your observed, right? This is observable. This is data. This is data from a sequence from today's time point. And you are looking at two chromosomes. All right. So if you're looking at two chromosomes, you are scanning the data from linearly from this, suppose you are scanning the data linearly from this end, and if there is a lot of difference, then you say that if there is no difference or very little difference, then you say that they have an ancestor which is not too far back. And these are like discrete trees, right? So at some point of time, this is hypothetically at some point of time, this segment is from an ancestor who goes farther back. Because there is a clustering of mutations. The two chromosomes are very, very different here. So the hidden Markov model essentially tries to find out the difference, find out the points here. So this is the background probability model. This is the observation that you have. It finds out where does this transitions take place. Where does it require to invoke that the ancestor is actually farther back in time. And when you do that, using the coalescent theory, you can actually do the calculation in such a way that you get a sense of what the effective population size of this was. So when the ancestor is here, then if there are many ancestors who are from this time point, many ancestors in around this band of time, then you say that this is the time point in the past when the effective population size of this population was actually very large.
This effective population influence the next generation.
The effective population influences the present generation as well. And your data, your genome, a single individual's genome has the history, the entire history written all over it, right? If you consider it in the entirety, the history is written all over it. Okay, so this is again conceptual. It works wonderful in simulated data. So people have done this, and they have simulated data using these coalescent models, and they see that it fits wonderfully well. So if you do the hidden Markov and try and find out the transition probabilities under a simulated data, it works wonderfully well. So people obviously would ask the question that does it really fit? Does it really fit to the history of human populations that we know as of today? So what they have done is they have taken YR1. One, A is a Yoruban who is an African individual. This is another. So the red and the green are two African individuals, and these are two European individuals. This is a Korean and a Chinese. Taken the whole genome sequence of these individuals and done this separately on that, and looked at what is the estimate of population sizes for this individual. So you take an individual and look at the estimate of population sizes. And what you find is that beyond something around 60,000 years, it's all same. Every individual's estimated population sizes are all the same, which means it's not separable. They are the same population. So this is like you live in Africa, it's the same population, right? So whether you are European today or a Chinese or African, your genome gives the same effective population size beyond 60,000 years. When you come after 60,000 years, you see that, look at the red and the green. You see that there's a rapid decrease in the population size of anything else but the red and the green. So there's a bottleneck happening. Each population is decreasing in size, but for Africans and for non-Africans, the decrease is much more rapid. And in a few years, there's a huge difference, and that's because that there is only a few individuals who have moved out of Africa. So the effective population size of today's Europeans and East Asians put together at this point of time when people are moving out of Africa, that is very small. And that's very, very clear here. It's very significant if you compare the African effective population sizes and the Chinese and the effective population sizes for the Chinese and the Europeans. And then if you come to the modern period, they exponentially just burst. So people have also shown that it's very, very reliable except for the last few years and for anything which is beyond this. So just by looking at one individual now, you can every individual genome has the same history written into this. So just if you look into your genome, you can actually get back to some of these regions of your genome which we have all inherited from our African past, right? There are small regions of our genome which are all from our African past. So all of our genomes have these small bits of histories, some of which goes back to our African pasts as well. Yeah, I sometimes feel it just fascinating to think that all of our genomes are written in those very, very similar ways, and it's all there. It's just ways and means to pick it up. All right. So in the title of my talk, I also talked about the future. So I'll talk about the future in a little bit. So what's the future of our excavation of our past? And that's also is testimonial of another amazing technological breakthrough, which is something which is called an ancient DNA. So this is 2008, and many of you might, this is not the first one, but many of you might have seen a big splash in popular newspaper where people sequence the nuclear genome of woolly mammoth. So all of you know what a woolly mammoth is, right? It's something which is akin to an elephant today, and all museums have these huge models of woolly mammoths built there. And one of the findings of this particular paper was that the woolly mammoth is. So how many kinds of elephants do we have in the world? Anybody have any idea in terms of broad species? African elephant and Asian elephants. Yeah. So African elephants and Asian elephants. And do you know that it's most likely true that the African elephant and the Asian elephant cannot have a viable offspring? It's most likely true. So they cannot have a viable offspring. Their chromosome numbers are also different. They're so similar, but they're actually different species. They're morphologically very similar. People would imagine a tiger and a lion to be much more different, but it's not so, or a chimpanzee or a human. So an African elephant and an Asian elephant are much more different compared to a chimpanzee and human. All right. So the woolly mammoth happens to be closer to the Asian variant of an elephant, not the African one. So people thought that the big woolly mammoth should be closer to the big African elephant. It's not so. It's much closer in terms of its genetic material to the Asian elephant compared to the African elephant. So how do you know this? How do you know what the DNA sequence of an woolly mammoth is? Because you do not see a woolly mammoth now. You cannot sample from an woolly mammoth, right? So how do you know this? A fossil is just too much, but it's something very similar to that. So what they do is, there are places which are like natural refrigerators in the world. And DNA is a wonderful, wonderful stable molecule, all right? Woolly mammoth used to stay in Siberia and all of the. So there are many bones of these woolly mammoths which are in many of the Siberian caves. And if you take material from the whatever the remains of that, and if you do a lot of purification, if you're really, really finicky about quality control, then you can actually find out the DNA of woolly mammoths. And that is something which is not hypothetical anymore. People have done this, and people are now doing this almost as a routine. When I move to humans, I'll talk about this individual. This individual's name is Svante Pääbo. Svante Pääbo is the director of the Department of Genetics, Max Planck Institute. And Svante is known as a Neanderthal man. Well, I mean, he calls him as a Neanderthal man, whatever. But the point is that Svante was a grad student studying immunology. His father was a Nobel laureate in physiology, and his mother was an archaeologist. They never married, but that was what it is. Savante grew up with his single mom. And his mom was an archaeologist. His mom used to go to different places, and he had huge interest in archaeology, no wonder. And in those days, it was possible when he visited Egypt with his mom, he actually somehow stole or whatever, he got a little bit of an ancient Egyptian mummy, and he moved back to Sweden. So when he was doing his graduate work in immunology, his daytime work was to look at immune profiles or whatever, blah, blah, blah. I do not know that. During night, when his supervisor wasn't there, he made his lab equipped so that he was trying to extract DNA out of this Egyptian mummy, all right? Ancient DNA out of this Egyptian mummy. So, he did pretty well in immunology as well. He graduated with papers in immunology, and then he wrote a single authored paper in Nature, which I think is DNA sequence of Egyptian mummies or something, right? And if you remember Alan Wilson, who did all this path-breaking work of out of Africa, etc., etc. So Savante was at that point of time contemplating to apply for a postdoc in Alan Wilson's lab. Alan Wilson's lab was in Berkeley, California, and he was contemplating that. He wrote this paper, and then he gets a letter from Alan Wilson, which says that, Professor Pabo, would it be possible that I visit your lab this summer? So it was kind of a role reverse. He was looking for a postdoc in Alan Wilson's lab. Alan Wilson tried to visit his lab, which he didn't have. So eventually, Savante went to Alan Wilson's lab, and there they tried to do this in a much more systematic way. Okay, so if you start doing it with Egyptian mummies, then the next thing you probably think about doing is looking at Neanderthals, right? So the Neanderthals, as we mentioned, were very, very widespread all across Europe. All across Europe, you see many, many Neanderthal remains. Actually, they derived their name from Neander Valley, which is a place in Germany. And there are, as you see, these are all Neanderthal remains, all right? And there is this cave in this place, which the caves are wonderful because the temperature within the caves, they remain kind of constant for a very long period of time. Come summer or winter, it does not change too much. So caves are wonderful to have preserved bones, preserved molecules. So what they have done is they got this from these caves, and when Savante started his lab in Germany, he started to look into this. Started to look into the DNA of this, and whether you can extract DNA from the old Neanderthal bones. And this is December. You might be seeing this in papers now that there are these breakthroughs of the year, right? At the end of the year, Nature, Science, and all of these magazines, all of these journals, they come up with what are the breakthroughs of the year. So now we have the breakthroughs up to 2018 and so on and so forth. So this was the breakthrough of the year 1997. And what they did was they got the first Neanderthal DNA sequence. All right? So they looked into the mitochondria of Neanderthals. They could extract the Neanderthal mitochondria, and they could actually sequence a small segment of it. Okay. So they could get 123 clones of 13 amplifications. Do not worry too much about that. So what they did is they could get 379 base pairs from the Neanderthal individual, right? So 379 base pairs from Neanderthal individuals, and then there was data at disposal. There were 478 Africans, 510 Europeans, Asians, Native Americans, and so on and so forth. So people from all other places, you have the mitochondrial data from all of them. So you compare with the Neanderthals and come up with the question. So this is another raging debate which we had. Go back to the multi-regional hypothesis. People have postulated that the Europeans are essentially descendant of Neanderthals, and then there are these Java men who are ancestors to today's East Asians, and that's the reason people had the multi-regional hypothesis. Okay, so when they did that, they compared the sequences. The drill remains same. You compare the mitochondria of a Neanderthal, compare the mitochondria of a human, look at the number of differences. Compare the mitochondria of the human, compare the mitochondria of the Neanderthal with the mitochondria of a Neanderthal, look at the number of differences. And what they found is that the modern human sequence at this small 379 base pairs differed on an average of at 8 positions, all right, compared to the 8 substitutions. And the difference between humans and the Neanderthals is 27.2. So there was a huge difference. So any two individuals, of all of these individuals that he had sampled, or that people have sampled from from different populations of the world, the average difference was around this, and the average difference here was this. So this is how the distributions look like. So compare a human with a human. This is and draw a histogram of the differences. This is how the histogram looks like. Human versus Neanderthal, the histogram of differences, this is how it looks like. It's clearly separated, right? It's a completely different thing. If you compare human and chimpanzee, then this is how it looks like. So there are many, many ways in which people have calibrated the difference between separation of human and chimpanzee from a common ancestor. And this is something around 5 million years, which is, for that particular region, is equivalent to something around 55 differences on an average, right? So 5 million years, 55 differences, how much will 25 differences account to? Simple unitary method. So people thought people could come up with the fact that Neanderthals and humans, they're different species, and they actually separated for a very long period of time. And irrespective of where you sample humans from, they're much more closer to each other compared to Neanderthals. So so it was already put to rest by all kinds of DNA data that came in, but the multi-regional hypothesis, again, is put to rest here, right? So great. I mean, this is 1997. This is a time when people used to use all kinds of old technologies. This is the time when if you had sequenced 500 base pair segments, then that was good. All right. So this is again. I mean, this is again the red is how the humans have moved, and the Neanderthals were right here. So now here is a question which is much more profound and which cannot be answered just by looking at the mitochondria. And the question is that, all right, so if you look at the mitochondria, they are like completely different species. They have a common ancestor, but they're completely different species, and there is a time difference between them. But there is a possibility that there is surely an overlap with humans and Neanderthals. So can we rule out the possibility that there might be mating between humans and Neanderthals? And the matings might also result in offsprings. And if that be so, then in the human genome, in today's human genome, we might find small regions of the genome which is akin to a Neanderthal, right? If there is mating which gives rise to offspring, then what will happen is, in the first generation, the offspring will be 50% human genome, 50% Neanderthal genome. Over time, if this individual mates only with humans, if subsequent generations the mating is only with humans, then the proportion of Neanderthal genome will decrease in size because recombination will take care of this. But still, if it has happened at some point of time, then it might still be true that occasionally, just accidentally, in the human genome, we might find small stretches which are like Neanderthals. If we find that, that means that human and Neanderthals were capable of having viable offsprings, right? Okay. So can that be even, that can obviously be hypothesized, but can that be tested? So that would mean that whatever was postulated before, which is when we moved out, anatomically modern humans moved out, we replaced everything. It might not be a complete replacement. It might be a little leaky. We might be, while we were replacing, we might be having a little bit of other people's genomes assimilated into us as well. So in 2009, because they had the DNA extracted from Neanderthals, you remember at 2007, there was this new technology which came up, which changed the entire paradigm. They actually used that technology to sequence the entire 16 kilobases of mitochondria. So they could actually have a reference genome for the Neanderthal mitochondria. All right, so this is 2009. In 2010, they could sequence the entire 3 billion bases, which is a draft sequence of the Neanderthal genome. Now, if you have a draft sequence of the Neanderthal genome, then your problem becomes much more easier. So you have this genome of Neanderthals. you have these genomes of humans, and you just go comparing between this and this, and if you see that there is this chunk of DNA which one individual have, one human have, which is very similar to a Neanderthal, but is very dissimilar to anybody else, very dissimilar to all other humans, then most likely this has an ancestry to the Neanderthals, right? And you can do this for all humans. If you have a reference sequence, it's easy to do. And something very similar to that was done, and then we came up with this surprise. So these are Neanderthals. So I mean, Neanderthals were supposed to be, were very successful, by the way. I mean, they also moved out of Africa, and they actually were very successfully spanning a very large geographical region, as you can see in the map. I mean, they were very successful, and they went through multiple waves of ice age and global warming, and ice age and global warming, and so on and so forth. But somehow around 30,000 years ago, they got extinct. This is after we moved out. I mean, and that's the reason people thought that it's all replacement. So what we found was pretty surprising. That you found that it's about 1 to 4% of genomes of people in Eurasia are all derived from Neanderthals. So it's 1 to 4%. That means you and me. That means all of us have something around 1 to 4% of Neanderthal genomes within us. And this is also, again, something where there's a very clear distinction between Africans and non-Africans. African genomes, individuals of African origin, do not have Neanderthal genomes. They have evidence of there might be admixture from other archaic hominids, but there is no evidence of Neanderthal admixture with Africans. But if you move out of Africa, any place in the world, actually, any other place in the world, there is about 1 to 4% of the genomes are Neanderthal. And it's pretty constant. And just imagine this. If my genome and your genome and all of our genome has something around 4%, it's fixed. It's not going to go away. Because if you pass on your genome, you're passing 4% of these Neanderthals, 4% of this Neanderthal genome to the next progeny. Because the person whom you're mating with and passing on your genome is also having 4%. So it's not going away. It's a part of you. There is no way we can even try and get rid of this. And why should we? I mean, actually, we are better off. And I might give you an example of how we are better off having a little bit of Neanderthal into us. Okay, so it bolsters the out of Africa model, because Africans do not have it. If you move out of Africa, you have it. And that's where there is also the entire archaeological and other kinds of evidence of Neanderthals living. So this is a place, if you move out, this is a place where there were Neanderthals, and there was mating between humans and Neanderthals, pretty extensive mating between humans and Neanderthals, and there were many, many people who are actually offsprings of humans and Neanderthals, and it has it's pretty widespread that way. Okay, so everybody else has it. So obviously, as you can imagine, this endeavor hasn't stopped. People have gone and getting ancient genomes from all over the places, from ancient human skeletons, and doing this, right? And they have come up with other kinds of surprises. There was recently a skeleton in Romania of a young girl, and when they did the genome sequence of this young girl, they found that this young girl, whose skeletal remain is about 60,000 years old or 45,000 years old, I forget, this young girl is 1/4 Neanderthal, right? Pretty clear testimonial of that one of the 4 grandparents of this Neanderthal was a Neanderthal individual. All right. But then there is something else. There is a twist in the tale of this story as well, which comes from a cave somewhere in Siberia, which is called a place called Denisovo. So there was a cave in Denisovo where there were many, many Neanderthal remains. So people now were curious that you have sampled the Neanderthals from places in Europe. If you sample from places in Siberia and compare between the Neanderthals there. So we are getting curious about the genetic diversity of Neanderthals now, right? Like we were curious about the diversity of modern humans and sampled modern humans from different parts of the world. we are sampling ancient Neanderthal genomes from different parts of the world. So people did it from Siberia in a place called Denisova. It's called the Denisovan cave, and came up with a digit from the left, this digit from the left finger, right, of a young girl. It's a Neanderthal bone. They went back, sequenced this, and found that the sequence does not match a Neanderthal. It does not even match a human, anatomically modern human. And it's a completely new species. And there is no way, even anybody speculated that there is another kind of an hominid who was living there. And they call this as a Denisovan. So there is very clear evidence of another hominid species from whom you know their DNA, but you do not even know how their skeletal remains would look like. You just have a small digit in from the left hand. So you just know how their left hand look like, but you do not know anything else. And it looks like a Neanderthal as far as that is concerned. It's not distinguishable otherwise. But if you look at their DNAs, they're strikingly different. And this species came to be known as Denisovan. And then you start scanning the interworld data, and you find that there are places in, so this is where the Denisovan cave is. There are places here in Papua New Guinea where you find a huge amount of Denisovan admixture. So not only was Denisovans here, the Denisovans were probably very widespread here as well. And the drill is now similar. So the people in 2012, you get a draft sequence of the Denisovans. You compare between all other populations, and you find that in Papua New Guinea, the Denisovan proportion plus the Neanderthal proportion, that makes about 6% of the genomes, which is, I think, astonishingly high. There might be other archaic hominids as well, but this two makes 6%. Somewhere back in this year, one of the contenders for breakthrough of this year in, I think, Nature or Science, or both, is that they serendipitously found another remain of a skeleton. If you look at that, that individual is a 50% Neanderthal and a 50% Denisovan. So from one of these caves, when they went on sampling, they actually found an individual who is admixed between a Denisovan and a Neanderthal. So yeah, I mean, the new technology, the new way of looking at this, and the new kind of data has put this thing in a completely different kind of pedestal. It's a completely different kind of a way in which people have started looking at this. And this map of movements all across the world has been defined and redefined in many ways. If you, I'd like to end this with, I think I do not have the, I do not have the slides which tells you how we have benefited from our admixture with Neanderthals and Denisovans, but believe me, for the time being, that we actually have. And some of the reasons why maybe we are even able to survive in today's environment is because of our admixture with Neanderthals. Maybe that's true. All right. So what I'm trying to emphasize in the 5 minutes excess of whatever I have been given is that there is some kind of a tree, and we think about human evolution as bifurcating trees, which are pretty simple to see. There are species have common ancestors, and species they bifurcate, and so on and so forth. It's probably not that simple. It has a much more nuanced and approach is probably required to look at this, because whenever this is like this, there is huge amount of huge possibility that there are exchange between these so-called species. So whatever was a definition of a species in classical biology, where species are like separation such that you do not reproduce to have a viable offspring, that's probably not entirely true. And there are many counterexamples in the world where people have seen that this is not entirely true. This is hugely true for what we consider is for the hominid species in general, right? So modern humans, anatomically modern humans, we today are essentially resultants of multiple kinds of admixtures that has taken place all over back in time. And this is and very recently after that, in the last, 50,000 years, more so in the last 12,000 years after advent of agriculture, etc., and the last 200 years when after the advent of industrialized revolution, etc., we have expanded in size. And all of these genomes have become integral parts of us. And it's not separable. And this is actually a wrong way of looking at evolution. It's not linear. It's not so easy. It's not that you start here and then eventually you get back to this, or even this. I mean, we can all make fun of all of this, but this is not how it works. It's not this. It's a much more complicated scenario. Just imagine this, that it's not too far back when this whole hominid thing separated from the big apes. And there are so many trials which have gone through. Chimpanzees, the two kinds of chimpanzees, the bonobos and the big chimpanzees, they survived, and we survived. And everybody else, some of them are present in traces into us, but most of them, they are just not here. So it's not like a linearity in which you can get from here to here. There are many, many missing steps in between, and many, many trials which were there. Some of these were very successful. Some of them have lived in the world way more than we have. In fact, most of them. So there is no time in which we cannot even say that we are as successful as these guys. I mean, we might get extinct much earlier compared to anybody of them. And it's no way a linear improvement kind of a thing. It's a much more complicated scenario than this, and probably genetics is getting us a sense, a handle to understand that way better than anything else had ever been so successfully able to handle these kinds of problems. All right, thank you for listening. So this.
Okay, so we have questions.